is Dr. Jack Auty, and he's a lecturer in medical sciences in the School of Medicine at the University of Tasmania. His research focus is how inflammation can contribute to disease. And Jack has a broad range of interests, from how microplastics induce lung and gut inflammation to whether commonly used anti-inflammatories can be used to treat Alzheimer's disease. Thanks for joining us, Jack.
Hi, how's it going? Can everyone hear me?
I can hear you, yeah.
Oh, that's great. Fantastic. Okay, so let me jump into it. Maybe I should start at the beginning of my talk, sorry. So if I hit share screen. Is that all good, Brad?
All good. Yeah, sure
nice.
Can.
Brilliant. Yes. So my name's Dr. Jack Auty. I'm a lecturer in medical sciences at the University of Tasmania. And today I'm going to be talking about the problems of particulates. So you've probably had a lot of talk, we've had a lot of talk so far about the chemical makeup of these contaminants, but I'm going to be looking at it much more as a physical problem, these particulates as a physical problem, looking specifically at microplastics as well as nanomaterials. Now, my entire research career has been about immunology, and I love the immune system. So I just thought I'd put this little video up here. These are my white blood cells, and they are doing one very cool thing that the immune system does, which is spewing out their DNA to trap bacteria, which is amazing. It's like using the most complicated molecule in the universe, DNA, to like a rope to trap bacteria. But that's just one way the immune system's really cool. Hopefully I'll convince you the immune system is further amazing throughout this talk. So what I'm really interested in is this concept, and I guess this whole conference is about this. It's about this concept of mind the gap. This is what I call it. Now, it goes a little something like this. Here's a timeline, and at some point in time, we have an innovation or an invention. Now, almost immediately after we invent something, we typically apply it. So it immediately goes out and is applied into our world. And then there's a very big gap in time before we realize the health and environmental consequences. And this gap is essentially caused by a lack of research, because we don't thoroughly research the application before we use it, and so we end up with this big gap. So I'll just give you one tiny example. You know, radium is a radioactive atom that was discovered in 1898. We immediately applied it 10 years later to make glow-in-the-dark watches, and they were painted on, this radioactive paint, onto these watch dials to make them glow in the dark. And then it wasn't until 50 years later that we stopped using radium paint in these watches, after about 2/3 of the workers who were doing this work to paint the watches had died of radiation poisoning or cancer. And there's lots of examples of this, you know, CFCs and the ozone layer, CO2 and global warming, asbestos and lung cancer, lead paint and neurotoxicity. We typically invent things, use them immediately, and then through really a lack of research, it takes us a long time to realize those health and environmental consequences. So I'm interested in this gap and trying to deal with this gap. And so with nanomaterials are these new groups of materials that we're producing, where we essentially can build them on a molecular scale, and they have all sorts of unique properties because of the way we produce them. And they are probably, I would say, around about here in the timeline. We have invented them and we are applying them with almost no knowledge of the health and environmental consequences of these nanomaterials. Whereas plastics, I think we're over here. We know it's bad for health and environment. Now the question is how bad for the human health as well as environmental health and health of animals in the environment too. Now, plastics, we're very familiar with the problem of macroplastics, like litter. Birds are eating litter, turtles, you know, have straws in their nose, and fish are getting strangled by those beer canister holders and stuff like that. So we're very familiar with the problem of macroplastics. However, microplastics we're not familiar with. These are the very, very small plastics that can't be seen with the human eye. What kind of problems are these causing in the environment and in human health? We're only just starting to get a grasp on it. And for me, you know, a lot of people talk about microplastics to mean different sizes, but for me, it gets really biological biologically interesting when they're below 20 microns, because at that scale, they can start to get inside of individual cells within the body. And that's when a whole bunch of unique processes can start to happen. So here are some microplastics here. How do we get exposed by microplastics? Well, there was recently a study published showing that a single tea bag can release about 11 billion microplastics into your cup of tea whilst being brewed. So every cup of tea you've ever had has billions of microplastics in it if it was made out of these fancy triangular bags. The square bags, I believe, are not made out of plastic. So look for the square bags, not the fancy, or do loose leaf. Another example is clothing and carpet. Every time you remove a synthetic jumper, there's an estimated 700,000 microplastics being emitted as you take that off and then you breathe it in as an aerosol. And several studies have pointed out that now a large percentage of household dust is made out of microplastics. So we're definitely drinking it and eating it. We're definitely breathing in these microplastics. What possible effects are they having on the body? Now, nanomaterials, the one that I researched is graphene as an example of it. And essentially, it's a single atom thick molecule sheet that looks like chicken wire made out of carbon. So this is it here. And it's essentially, you could imagine it as a single layer of graphite. In fact, the first time they ever produced it, they produced it by taking a bit of graphite, which is like pencil lead from your pencil, and putting a bit of sellotape onto it and peeling it off. And it would peel a single layer of carbon off. And it has a whole bunch of unique properties with many potential applications. It's stronger than steel on the molecular scale. It is a better conductor of electricity than copper. It can catalyze reactions. They've put it into concrete and shown that if you put graphene into concrete, it now soaks up more CO2 from the air. And we're even using it in the medical industry, and this is where I got involved. We're potentially looking into it as a drug delivery system or as a delivery of things like RNA or DNA into cells. And that's where I got really interested into it. So given all this mind the gaps issues we've had in the past, you'd probably think we're really careful with these nanomaterials that we've just started developing, and we won't be applying them until we've done thorough research about potential health implications. Well, that's certainly not true. You could go out and buy a shoe full of graphene right now for some reason. I really don't know how it could improve a shoe, but apparently it's been used in shoes. And they've even started to research it as a way to produce, essentially to put RFID tags on food, a way to create electronic circuits in food. So we're certainly not being careful with it already. So my questions are, what are the health consequences of these plastics, particularly microplastics, as well as the nanomaterial graphene? And I had the hypothesis that these materials will cause chronic inflammation, which is negative health consequences. So I'm just going to give you a little snapshot of inflammation. So here we have a piece of tissue with regular cells in there, and here we have an immune cell sitting in that tissue, and here we have a blood vessel going on. So if you were to get tissue damage, we would literally rip open our cells here, and compounds that are within the cells will come out of the cells, and those compounds that should be inside of cells will be detected by our immune cell there. Another thing is you might have a pathogen like bacteria, and they will release compounds that can also be detected by our innate immune cell there. This immune cell, after detecting it, is literally smelling the extracellular environment, using receptors to detect anything. They will release a range of inflammatory signaling molecules. This will cause vasodilation, which means swelling of the blood vessel, and leakiness, which means fluid will flow into the tissue. And this is why your ankle swells up when you roll your ankle. But it will also recruit hundreds of immune cells to the site of injury or infection, and these immune cells will do a couple of interesting things. One thing here, we have a neutrophil. It's spraying essentially toxic chemicals into the environment to sterilize it. One of these toxic chemicals is bleach. It's chemically identical to bleach, and this bleach is to sterilize the environment. And another thing these guys do is phagocytosis, which means to eat up the cells in the environment. Now, if either of these processes are excessive or chronic, that can cause a huge amount of problems with disease and tissue damage, because you imagine a lot of bleach in your tissue for a long period of time is damaging. If we were to imagine this was the lungs, you can imagine having bleach in your lungs, having immune cells eat your lung cells, and having fluid rush into your lungs chronically is a very bad thing for your lungs, right? And so the one question might be is what receptors, here we've got these receptors, what receptors might be involved in plastics and nanomaterials? And this is something I'm interested in. And I hypothesized it was this receptor. This is called the NLRP3 receptor, and this receptor detects stress within the cells. And that can be physical stress, such as being poked or prodded with a very small particulate, or perhaps the immune cell has eaten a particulate that it cannot digest. So it phagocytoses it and it cannot digest it. So it ends up with phagosomal stress, which is almost like a bellyache for the cell. Now, when this receptor is activated, it's a beautiful receptor. It releases one of the most inflammatory, probably the most inflammatory signaling molecule in the body called IL-1. And this is just an inflammatory protein that signals for all those things, vasodilation and immune cell recruitment. Now, this the reason why I suspected it would be this receptor and this signaling molecule that's involved in this pathway is because of the diseases that we know that this receptor and signaling molecule are involved in. For example, gout, which is a buildup of urate crystals in your joints that cause huge amounts of inflammation. We know that that is signaled via this pathway. Silicosis, which is the breathing of mineral dust, which miners go through, causes massive inflammation through this pathway. Asbestos, that spiky insulation material, also causes it through this pathway. Cholesterol crystals, and you'll see here something in common with all of them. It's particulates. It's an asbestos particulate, a urate crystal particulate, a cholesterol particulate. Even Alzheimer's disease is a protein aggregate particulate is one way to think about it. This is a form of dementia in the brain. And this receptor and inflammatory signaling molecule has been implicated in all of those diseases. So the question is microplastics and nanomaterials also an activator of this receptor? And does it similarly cause disease like chronic activation of this pathway does in all these other diseases? And so I did a Friday afternoon experiment where I took a nail file to a tip box and I put it on immune cells and I measured the inflammation, the inflammatory signaling molecules coming up. And what I found was that the plastics induced a lot of inflammation. Here's my positive control silica. And I also had immune cells that had this receptor genetically deleted from it, and we got no release of the inflammatory signaling molecule. So we can immediately see that plastics, microplastics are inflammatory. Bit of a dodgy experiment. I think we've all got to admit that. And but it is NLRP3 dependent. So I wanted to get actual forms of microplastic. I didn't want to get these beads that you could buy. I did buy those beads that you can buy from Sigma, and they weren't inflammatory, which was very confusing. So I wanted to get, I felt it was to do with the shape and my method of generating microplastics. So we actually went to a facility that generates parts for spaceships and airplanes because it had the ability to mill plastic at the micron scale. So here we have a strip of microplastic being milled, and these dust particles that are coming off, she's just putting into plastic. We wore masks when we collected it, I promise. And we collected, this is my PhD student, Barley Lee, and we collected those plastics, and then we put them on macrophages. Here they are here. And what we found was, yes, nylon, polyethylene were very inflammatory in these macrophages, but polystyrene wasn't, which was really interesting, actually. It does depend on the kind of plastic, how inflammatory it is. And we showed again by genetically deleting this receptor, this inflammation was dependent on this receptor here. And so we did a scanning electron microscopy to figure out maybe it's something to do with the shape there. And we did notice that, you know, there is a difference in the structure of the microplastics when generated through milling. So the nylon here and the polyethylene form these more spiky, elongated with high eccentricities, these particulates, whereas polystyrene produced a more rounded shape there. So perhaps it is related to structure, and the more spiky crystallized crystal spike looking like plastic is more inflammatory. And we did in vivo experiments, a number of them, but this is just a really visual way to see it. And this is an injection of the microplastics, very low dose actually, of microplastics under the skin of mice. And then you can flush the area under the skin 24 hours later to see what cells are present under the skin. And in the control group, this is normally what you would see under the skin of a mouse. And this is after an injection with nylon. And you can see it's absolutely full to the brim with immune cells as they rushed to the site of this microplastic exposure going on. So it is relevant in vivo. We did a number of other experiments, but I don't have time. I've got to get to the graphene as well. But another little side note is we actually got plastics from the environment. But this is because plastics undergo changes in the environment thanks to UV exposure. They also soak up heavy metals and stuff like that. So this is a little teaser about where my research is going. We showed that environmental microplastics were actually more inflammatory than the naive microplastics, the most inflammatory naive microplastics I had discovered. So we're delving into this area right now. We're very interested in it. Now onto the graphene. Here's this graphene. It looks like chicken wire made out of carbon. Here's an atomic force microscopy image of it. Now, we were going to use this to deliver RNA and DNA. The people were investigating it as a delivery mechanism for things like RNA, which people are very interested in. And we actually used this thing called graphene oxide, which is just an oxidized version of graphene. And what we found, so in black, we first wanted to see, is it inflammatory? So in black here, which you can't actually see, we just checked whether graphene oxide was inflammatory, and it wasn't. So this is immune cells in a dish given graphene oxide, and it wasn't inflammatory. So then we checked, does it maybe exacerbate inflammation? We thought if we initiated the inflammation, adding graphene oxide would exacerbate the existing inflammation. And what happened was, it inhibited inflammation. So we ended up with this really perplexing result that this nanomaterial was actually anti-inflammatory. And it was sort of the opposite of what we expected, because we expected the particulate to activate the particulate inflammatory response system through this NLRP3 receptor and IL-1. And but we thought, why don't we check more than just IL-1, the signaling molecule? So here's the IL-1 signaling molecule. Here's another result here showing that you give an inflammatory stimuli, you give graphene, you inhibit the inflammation. But here's another inflammatory signaling molecule. This might be the second most important inflammatory signaling molecule in the body, TNF-alpha. And we showed that graphene actually increased the inflammation in TNF-alpha. And this was very confusing. And these techniques we were using was essentially a method to investigate each gene one at a time. It's called qPCR. It allows us to evaluate which gene is switched off and switched on one at a time. And we decided that to really understand what was going on, we needed a bit more big data. So we used RNA-seq, which is a way to look at all the genes and how the genes are dialed up and down in our immune cells. And what we found was there was this interesting dichotomy. So red means upregulated, blue means downregulated. The graphene oxide upregulated half of the inflammatory genes we looked at, and downregulated half the inflammatory genes we looked at, which was very peculiar. I don't want to go into this, I don't have time, but we investigated how these effects might be going on, and what we discovered that graphene oxide had massive effects on the metabolism of the immune cells. It promoted glycolysis, and it blocked sort of the electron transport chain processes going on. So it seemed like it might be damaging or turning down the mitochondria in the cells. And we figured out how this molecularly related to its inflammatory nature. But a really important point there is we wanted to know, these were cells in a dish, we wanted to know which pathway, the inflammatory or anti-inflammatory pathway, is more important in vivo. So we did the same sort of injection experiment under the skin of mice, and we found that graphene oxide was actually massively inflammatory when you inject it under the skin of mice, and this seemed to be through the NLRP3 receptor. When we gave an inhibitor, we blocked it, and it also seemed to activate other inflammatory pathways and other inflammatory signaling molecules. So this is, sort of one of the reasons why we need animal experiments, is cells in a dish don't replicate animal models. Thank you very much. Sorry, Brad, did I make it? Thank you very much for listening. If you have any questions, flick me an email.